Introduce myself. I'm Francesco Berti from Novartis Vaccine, Italy. So today I'm going to talk about some which appear as a sort of classical technology for preventing meningococcal infection. And this is substantially a new target which is substantially emerging in a specific region in Africa. So we already had the opportunity yesterday to have a brief look on the epidemiology of the meningococcus X, which is substantially emerging in some specific area in Africa, particularly in sub-Saharan area, and where we have mainly three different serogroups which are causing the majority of infectious disease cases of meningococcus, which are substantially the meningococcus A, the meningococcus W, and meningococcus X, which is obviously emerging. While for meningococcus A vaccine, a monovalent vaccine has been already delivered in that area, which is substantially the product developed by the meningitis vaccine project in the context of PATH, and many doses are already available for people there. And also for meningococcus W, which is substantially one of the serogroup included in the tetravalent vaccine, commercially available by several companies such as Novartis, GSK, and Sanofi. And for meningococcus X, unfortunately, no vaccine is available at the moment. But the capsular polysaccharide, which is substantially covering the bacteria from this serogroup as for the other groups, is substantially a very good target that could be used as a sort of potential candidate to be developed for a vaccine against meningococcus X, as a monovalent vaccine and also as a combination vaccine with the other serogroups, mainly circulating over the world, which are A, C, W, Y, and recently the meningococcus B. Substantially, the technology of conjugate vaccine appeared to be really old, but so substantially this was developed at the beginning of 1980 years by the group at NIH of Professor John Robbins, but it still remains probably one of the most promising technology for developing conjugate, for developing antigens for many pathogens. And the pure polysaccharide, unfortunately, are not really immunogenic in young children. We have many reasons just to move directly to the conjugate vaccine, which is the most promising. Substantially because we can just have several advantages, such as immunogenic and protective effect in young children, T-cell dependent immune response, booster effect, no hyporesponsiveness, and substantially a switch from IgM to IgG response, also in the young subjects. This is just a brief overview of the two current technologies that could be used for polysaccharide, and this is just the mechanism of actions that characterize both the plain polysaccharide and the conjugates. So this is just the mechanism of action of the polysaccharide-based vaccine. We have the B-cell receptor, we have just the substantially processing in the B cells, we have no production of B cell memory, and substantially we have a mainly production of IgM and no switch to IgG. When we move directly to the classical mechanism of action of the conjugate vaccine, this is something published in 2009. So in 2011 and recently other papers come out from the literature revisiting the mechanism of action. This is something really classical because it seems that there are other aspects that I don't touch today. But at the end of the day, we have just the same B-cell receptor. We have an internalization of both components, the protein and the polysaccharide plain polysaccharide. We have substantially a processing of both components in the B cells. We have a re-exposure to the MHC class 2 and substantially a T-cell receptor, which substantially recognizes the peptide component, as in some specific cases this is part of the discussion in the scientific community at the moment, also for the polysaccharide component, because substantially the glycopeptide is re-exposed on the MHC2. And that the results of this kind of behavior is just production of IgG and IgM, and substantially many of the advantages that I already listed in the previous slide. The success of the conjugate vaccine has been collected over the last 25 years approximately. So because the first vaccine was essentially licensed, the conjugate one was essentially licensed at the end of 1980 years. was a vaccine against Haemophilus influenza type B. And after approximately 10 years, the UK started a very large campaign of immunization for many subjects, ranging from 0 to 16 years old. And this was a success, because if you look at this picture, this was just the proportion of cases, the number of cases. Please have a look on the orange part here, because this is just the case of B and the other serogroups. But if you look at just the part B of this diagram, you can see that after 5 years, or approximately 5 years from the first immunization, which occurred in October 1999, the cases of meningococcus C dropped dramatically, and was a great success. While for the other serogroups, unfortunately, no protection was obtained. We don't have cross-protection with the other serogroups. We have, unfortunately, the polysaccharide, which specifically cover the specific serogroup infection. And moving to something that could be related to the chemical structure of the new polysaccharide. This is just the chemical structure of the meningococcus X, which apparently seems to be really similar to the meningococcus A, as our colleague from the Indian Institute mentioned yesterday afternoon. So this is substantially a glucosamine, an acetylglucosamine with a phosphate group in position 4. This is substantially the repeating unit of this polysaccharide. And we recently published also some theoretical knowledge to better understand the characteristic of this molecule. And the second ingredient for making the conjugate vaccine is the cross-reacting material, CRM, CRIM, just to use the short acronym, 197. And this is a very well-defined molecule, because just mutating this amino acid in position 52, you can completely detoxify the diphtheria toxin, which is substantially the source of this material. And this protein is naturally detoxified, because substantially we don't need to use chemical treatment, as is obviously requested for diphtheria toxin or tetanus toxin to produce diphtheria toxoid or tetanus toxoid. And the main advantage of using this protein is that we can easily prepare the materials for immunization. The conjugate vaccine is very well defined. We don't really need just to have many coupling chemical linkages, and so that's much more convenient, also in terms of consistency in the GMP production. Some more details about the preparation of this conjugate vaccine. Substantially, the polysaccharide is obtained by bacterial growth and harvesting the capsular polysaccharide, which is mainly released on the supernatant of the bacterial growth. We have several steps for purifying the sugar, steps for protein coupling, and finally the purification of the final conjugate. So, in the process for preparation developed by Novartis vaccine, we got a lot of experience for using chemical treatment, not using enzymes or other reagents such as phenol, just to extract and purify the polysaccharide. And also, the yield was comparable to the current productions of ACWY, which is one of our vaccine already licensed. And secondly, the level of purity was really good, just in comparison to the other polysaccharides already licensed, in terms of residual content of protein, nucleic acids, and endotoxins. The fingerprint of the NMR spectrum confirmed the identity of the chemical structure and the conformity of this structure along the entire production process and the different intermediates. Two different conjugates, substantially this was the work published on PNAS last year. And so two different conjugates were prepared in order to be tested and in order to design the best candidate for potentially for clinical trial. using two different linkers, so a longer and a shorter spacer, just which is the component to couple the sugar, the carbohydrate antigen, to the carrier protein, the T cell epitope here, which is CRIM197. And at the end of the day, so both conjugates were prepared in a very good yield, and just having a sort of final construct, which is something represented here. I hope that we start. So this is the structure of CRIM197. In the blue highlighted, we have the lysine residues, and the red is just the lysine which are used for coupling the sugar. And finally, we have the structure of the sugar, which is substantially linked to the carrier protein, but using a terminal end. And this is much more convenient because you can obtain a very well-defined product in comparison to the cross-linked structure, which is substantially obtainable using a random chemistry around the saccharide chain. Okay, so some other details about the characterization in terms of size, in terms of saccharide-protein ratio, which is one of the critical attributes for inducing immunogenicity for this kind of antigen. So this is an HPLC profile just assessing the molecular size of CRIM197 before conjugation, and so the size of the two conjugates prepared by using two different spacers, the longer one and the shorter one. And also the SDS page, which is substantially the protein, which is very well defined, as I mentioned before, because it's just a single band. And after conjugation, you have the classical smear of the glycoproteins or glycoconjugates, which appear as a sort of not very well defined population, but it appears very well in the HPLC as a very narrow peak, not really complex in terms of preparation. In terms of weight per weight saccharide protein, here we have approximately 0.3 in terms of weight per weight, which is substantially not really good for chemical people like me, because I like just to use the mole per mole ratio instead of weight per weight, but this is the history for conjugate vaccine. And just to be really short, this is the classical weight we obtain for conjugates. The level of endotoxin was really, really low in the forend, pretty acceptable for any possible further development. More science. This is just something apparently not really related to the product development, but this is something useful to better understand how we can deliver this vaccine. One of the main issues for meningococcus A is the stability of the intrinsic stability of the polysaccharide in aqueous solution. And this is one of the reasons why our company developed a lyostate single vial for A and just a liquid for C, W, and Y serogroup. So this is the structure of meningococcus A. This is an N-acetyl mannose-amine. Just please have a look on this part of the saccharide ring. You have the N-acetyl group here, and this is just the degradation mechanism we published a couple of years ago. But if you change the orientation of this group, here is axial, and just please have a look here for X. You have the position equatorial here. But it's exactly the same thing for all the other linkages, except for the continuing of the polysaccharide chain, which is in position 4 for X, in position 6 for A. But so just changing the orientation of this group here, you can obtain a very high stability of this molecule. Because if you look at this profile, this is just the trends for meningococcus X, both at 37 and 45 Celsius, and this is the degradation pattern for A, just in liquid solution. I don't like to say that the A is not stable in liquid, in lyostate, because it's absolutely stable. This is just a sort of pilot model to compare the stability of these two molecules, which is the final result. So we can easily deliver this vaccine also in the meningococcus X, also in risk conditions such as where the cold chain is not very well maintained, such as in Africa. Another point, just discussing about the development of this product. You have a lab just which is able to synthesize molecule, small construct, and this is just the synthesis of the monomer of meningococcus X. This is a very useful compound for developing the analytic, for quantifying the sugar. And this is just an HPLC estimation using the monomer synthetically obtained in our lab that could be used as a sort of standard for building the calibration curve for quantifying the sugar. Again, we have the NMR profile confirming the comparison of the monosaccharide released by chemical treatment for a natural polysaccharide and the monosaccharide synthetically prepared, which is used as standard. So, which is the final message from the two last slides? Substantially, we have very good science in our team just to develop vaccine, but having a very nice approach to better understand which is the science behind. So, moving to the final part, just how this vaccine, this candidate, perform in the preclinical model, mice model. And we immunize mice using this dosage at 1 micro in terms of saccharide content, using alum phosphate as adjuvant, just testing the vaccine in bulb C mice, 3 doses subcut. The readouts were ELISA, just to quantify the total IgG content, and the functional response using the serum bactericidal activity with rabbit complement to understand, to know more about the functionality of the antibodies elicited in mice. 3 doses a couple of weeks apart. So, both conjugates, with the shorter spacer and the longer spacer, elicit very high IgG titers, both at post-2 and post-3, and also a very good response in terms of SBA, which is substantially very good at post-2 and post-3. Probably we are at the saturate level here, but so what we understood at that time, that probably just trying to understand which was the best candidate. Using a longer spacer, we obtained an higher IgG response at post-2 and an higher SBA titer at post-2 again. So this is just the preclinical work. Probably this is not a general rule because this is a mice model. How easy is to predict the behavior in human? Because in our previous experience for ACWY, we surely collect data which confirm very high response using adjuvant in the mice model, but the final vaccine license in human is without adjuvant, because the human is not a naive subject. We have pre-existing response, we have many other attributes to be considered, but this is just to select, and this is the model in our hands at the moment. So I would like just to briefly summarize what the key points I touched over the last 20 minutes, and substantially a vaccine against meningococcus X seems to be a real medical need, specifically in some developing countries. The epidemiological study probably at the moment are not really sufficient to guarantee the monitoring of the serogroup X cases over the world, but so probably we have just to improve the epidemiology, the tools for epidemiological evaluation shortly. Secondly, a candidate seems to be really promising based on the previous experience on the other serogroups of meningococci, and seems to be a good candidate for moving to phase 1 clinical trial. And so, as my final point, I would like to thank part of the co-workers of my team in Siena, in Italy, and you for your attention. And so I'll do my best for answering your question if you have. Thanks. Yep.
From epidemiological point of view, it would make sense to combine X with A, and why are you planning to do that? And for the Western countries to combine B with Y and C, to have this a little bit more targeted for the different countries.
Definitely. So I strongly support this kind of proposal because this is one of the best idea for preparing regional vaccine. Regional means so specifically a trivalent for Africa, because it seems that A, X, and W are the measured serogroups circulating in that area, and no other cases of C and B. And another combination vaccine that could be potentially really useful for the developed countries such as Europe is B and C together. So having all the components means conjugates in our hands, I think that we can easily develop the formulations just to fix the different markets and the different needs. Yeah.
Did you try to any other protein as to conjugate with these polysaccharides instead of that CRM197?
So we use also tetanus toxoid and diphtheria toxoid, which are substantially two common proteins used for testing the other serogroups, meningococcal. But so the best candidate in our hands still remain CRM197 because as I said at the beginning of my talk, this is a very well-defined molecule, and in terms of immunogenicity, probably it doesn't result in a great difference, but in terms of manufacturing, it's really much more convenient. This is my personal opinion. I strongly support this idea because, so being in a context of very complex molecule, I personally would like to guarantee the consistency batch to batch every time.
And how much the free PRP form which you get in the CRM197? Have you tried to that? How long it is stable in the conjugation form, or there is some free form of the polysaccharides after some time?
No, it's absolutely stable because the linker is much more stable than the phosphate group, the diphosphate group along the chain.
Did you use any stabilizer?
No, it's absolutely stable without any additional stabilizers.
Thank you very much.
You're welcome.
Thank you.
Thank you.